Welcome to the third of these Center for Multimodal Neuroimaging talk series. We try to do these every other month, and then in the months that we don't have these, we have a sit-down walk-up session in usually Building 10 in the atrium where you can come ask us questions about how to do multimodal imaging or how to do any kind of imaging, and usually one of us will have an answer or at least provide you with 10 more questions that you hadn't thought about that then you can go ask other people about. This month, we have Travis Riddle, who is definitely not a former spy, presenting on open science variation. And this was sort of spawned off of a talk in the summer series that was focusing on all the things you should be doing in your science in terms of making it open and transparent to the community. And with that, I will turn it over to Travis.
Thanks, Pete. Okay, so as Pete said, I'm going to talk to you about kind of the varieties of science and the different things that we might be interested in doing in order to make sure that we're doing the best science that we possibly can. I'm going to talk to you about a lot of things today. Most of this is work that other people have done, so none of this is my research per se. That said, I have done some work in this area, and I'm interested in these ideas, and so I'm kind of doing my best to synthesize a lot of different things that people have done, and I'm synthesizing them in a way that makes sense to me. So it may not, hopefully it also makes sense to you, but I'm happy to clear things up where necessary. Okay, so just to give an overview of where I'm going to go today. So I'm going to start off by giving you, I'm trained as a psychologist, so I'm going to give you a little kind of mini history lesson featuring some actors in the history of psychology. And basically, I'm going to use this to illustrate that in some ways, science has been static for a long time, right? There are features of science that have not changed. But in other ways, there are features of science that are dynamic. That is, they've been changing longitudinally. And one of the things that I think is important here is that the static bits and the dynamic bits don't necessarily fit together in the way that they always have. I'm going to talk to you about the kind of consequences of these static scientific structures paired with the dynamic scientific research programs that we all engage in or hope to engage in. I'm going to address a little bit the fact that science is extremely heterogeneous, and because of that, if somebody tells you should do X, like X may not necessarily make sense for the type of research that you do, but that doesn't mean that there aren't other things that you can be doing that could make what you're doing slightly better. Because this talk was kind of prompted by discussions around sample size, I'm going to talk to you about why sample sizes are typically important. So this is one feature of things that you might choose to do to make your science a little better. That doesn't necessarily mean that sample sizes are always the thing that you need to be focused on, but I think for large swaths of science, that is the case. And then I'm going to kind of end with this framing of the idea that open science is really, you can think of these practices as kind of technological advancements, right? That's not typically the way that we would think about a lot of the things that go on in open science, but insofar as technology is a set of things that we use to improve the state of knowledge, like I think a lot of the things that have been going on in open science and kind of ways of approaching science kind of fit that description. So just some notes about the language that I'm going to use. I'm often going to refer to things dichotomously as true and false. Insofar as we're biological or social scientists, like my personal view is that the state of the world is not so dichotomous, that things exist on a continuum. In fact, things are never going to be absolutely false, no matter what you're studying. It could be a very tiny effect size that's maybe not worth studying. But just kind of for the sake of communication and clarity, I'm just going to use this kind of dichotomous way of thinking. When I talk about previous efforts to replicate research, there's a lot of different ways you can use to assess whether a finding has replicated. And there's some active work going on in this area in terms of trying to figure out what a good way of measuring this is, but it's not totally settled. So if I say that like they ran 10 studies and X% of them replicated, I'm basically just plucking whatever they said out of the abstract, and understand that depending on which metric you choose, that number could be scaled up or down. So there's also some confusion around kind of the difference between reproducibility and replicability. So today I'm going to try to use the term reproducibility to refer to the practice of duplicating the results of a prior study using the exact same material. So this would be equivalent to like somebody sent you their data and their code, and you ran everything, and you got the exact same result. That's reproducibility. And you can contrast that with replicability, which is you're effectively duplicating the results of a previous study using the same procedures, but you're gathering new data. So you're basically taking what they said in the manuscript, rerunning the study, seeing if you get the same result. And it should be noted that reproducibility is basically a prerequisite of replicability, right? If I keep running the same set of scripts on the same data and I get different results every time, then that precludes anybody else from replicating what I've done. And so the first is a necessary condition for the second. Okay, so just
back up,
sure.
I'm confused. Duplicate the results using the same materials. The data is a material.
Yeah, and this.
So it's just reanalysis. It's not really reproduce. It's reanalysis of the data.
Reanalysis, but there are also occasions where there may not be data per se. So like if you're running like a simulation study and you set some parameters.
But it is, the thing is there and you are looking again at it. Why not reanalysis? Reproduce really complicates. That's okay.
Sure, you're right. I'm not like particularly beholden to these terms.
So
These are
instead of reproduce, say reanalyze?
I think reanalyze would usually be the same person. So reproduce usually.
No, same materials.
Yeah, I mean, that's a good point. Like, there are other ways of kind of framing these issues, and I just kind of picked the two that are most commonly used when talking about these things.
I've never heard reproduce with no new materials.
The
Right.
phrase is frequently computational reproducibility is becoming common in the literature. And that if you give me all of your data and your code, that I can get the same results as you want.
Yeah, I just call that reanalysis, but maybe that is reproduce because you're using the same code.
Yeah, but it's surprisingly rare that it is the case. Yeah. But yeah,
That's a
so it's just a
different
question of terms. Yeah, you need to stay on the mic.
But what if I see. If I use the same data but with a new software, I'm replicating?
Or-
It's a little bit squishy.
I would say that's, I mean, again, this is not settled like ideas, but I would say that's closer to reproducibility. So, and this a problem could occur here wherein you're using a different version of the software or you're re-implementing the exact same algorithm that somebody else did. Like, I don't know, is that a reanalysis, reproducible? The point is that I'm just trying to draw a distinction between cases in which you're basically collecting new data and cases in which you're taking whatever already exists and rerunning it. So reanalysis, fine with me, but I tried to pick the things that represented what you would typically find when people are talking about this.
So reproduce, no new data.
Typically.
Create new software. Replicate new data.
Yeah, that's a good rule of thumb.
Got it.
Okay, so brief psychology history lesson. So these two characters on the left is B.F. Skinner. I actually don't know what his real first name is, and Edward Tolman. So these are both behaviorists from the middle part of the 20th century. And behavioral psychology is generally strictly limited to just what you can observe about the entity that you're studying. So in Skinner's case, Skinner was what we would call a radical behaviorist. That means that like any internal representations that you posit to the organism is anathema. Like that is, Skinner did a lot to move our science, to move psychology beyond the kind of days of Freud and his colleagues. But one of the key aspects of Skinner's approach to psychology is that there is no internal states. The way that Skinner typically ran his studies was he created this thing called a Skinner box, put a rat or some other organism inside the box, and he basically looked at what the rat did when you presented it with different stimuli and schedules of reinforcement and schedules of punishment. He published much of his work in a series of books. One of the early ones is this Behavior of Organisms, and this, unlike today, when often when a scientist publishes their work, if it's scientific work, it's typically in a journal. If they write a book, that tends to be more for the general public. At this time, it was not at all uncommon for scientists to collect kind of their theory and a lot of their data, put it in a book, and that was meant for their scientific colleagues, their peers. So that, at least in psychology, would be pretty unusual today, and maybe in your fields as well. In contrast to Skinner, Edward Tolman he was also a behaviorist, but he was what was sometimes called a purposive behaviorist. I have a really hard time with that word. But effectively, what he ran one really nice study that I think illustrates some of the issues with what Skinner had believed. So he put a rat in a maze with nothing in it and just let the rat explore the maze for a while, right? According to radical behaviorism, this rat shouldn't be learning anything because there's no rewards, there's just a bunch of stimuli. After the rat had some opportunity to explore the maze, one day he puts a treat, a pellet, a reward at the end of the maze. The rat's wandering around the maze and realizes suddenly there's a reward at the end of the maze. The next time he puts the rat in the maze, the rat goes directly to the end of the maze. It's immediate learning, basically. And it's really hard to reconcile that observation with this idea that you're not learning anything unless there's some schedule of reinforcement and punishment. So this was a real problem for some of what Skinner was doing. Ultimately, psychologists came to understand that indeed you do have to it's hard to have a study of behavior without positing some kind of internal representations. And so this kind of debate went on for decades in the literature, and eventually we figured out that indeed it is the case that we need to have some kind of internal representation. Tolman also published a lot of his work in these same kinds of books. So also it's not really clear what Tolman thought about how women behaved. As you can see, this is definitely from an earlier era, right? This is not, in some ways we can look at this science and see, okay, there's a lot of similarities with what we do today, right? We have competing theories, we run experiments to collect some data that we think addresses those questions, and then we publish it, right? And we want our peers to see what we're doing. But there's also some aspects of this that are slightly different, like the book thing. In addition to the way that we do science, the kind of structures around science have changed in certain ways, right? So Walter Mischel was in my department when I was a grad student. He's died a year or two ago, but he would often tell these tales about what science was like when he first started in the field. He got his PhD in the 50s, and effectively who knows, maybe this is kind of apocryphal? Yes, that's the word I'm looking for. This could be a little bit apocryphal, but the story he liked to tell was that as he finished the end of his PhD, he sent off a half-finished dissertation to the University of Colorado. A friend of his advisors read it and was like, oh, this looks good, let's give him a job. And that was basically the only place he sent in his materials. Similarly, when he first applied for funding, sent in a reasonable request for funding and got it. Didn't spend a lot of time on either of those two things, but was assured of funding and a job. Any of us that have been working in science for the last several decades will certainly recognize that as being almost unrecognizable, right? That's like a completely different world than what we live in now. And this is backed up by data. So this is a slide taken from a talk given by Carl Bergstrom just like a week ago. And this is, the success rate of R01 funding since 1961. So in 1961, you send in for a grant, and if it's a reasonable grant, you have better than the coin flip odds of getting funded, which is pretty good. In contrast, I think this goes up to 2014 or so. At that point, funding rates are closer to 20%. And of course, there's variation here too, right? So Carl pointed out that some of his colleagues in biology, their funding rates are more in the kind of 5 to 10% range, which makes it hard to count on your ability to continue to produce science. Not only have funding rates decreased over time, but even the decision about how to allocate that funding has some issues. So there was this kind of heroic effort a couple of years ago where these researchers did as best they could to to mimic exactly the NIH grants panel experience. So what they did was they got 43 reviewers. They brought them all to the same place, same room. They pulled these reviewers from people who had received an R01 in the past 4 years, and they gave these reviewers 25 applications. Those applications they culled from previous NIH submissions. 16 of those applications were funded initially, and then another 9 were funded after revision and subsequent resubmission. So we have some that were obviously good, at least insofar as they were funded, and some that needed some work. So they looked at the agreement among all these cats in the room, and if any of you have sat on a grant panel before or maybe submitted a grant, you would probably not be surprised to learn that there's not much agreement among the reviewers. So this figure shows 3 different measurements that they took and 3 different ways of computing agreement for each of those measurements. And I want to say that it's funny how close to 0 the agreement is among the reviewers. In some ways it is kind of funny, but in other ways it's quite sad. And so in my opinion, rather than low agreement among reviewers, they should have just retitled the paper as no agreement among reviewers evaluating the same grant applications. So even aside from funding, there's some other structural issues in science that have changed. So it's no surprise to anybody in this room probably that there are an increasing number of PhDs being produced every year, but the number of faculty slots has remained more or less static. So in 1982, up until this plot goes to 2011, there's about 3,000 new faculty positions in sciences and engineering annually. In 1982, there were 19,000 new PhDs produced, and in 2011, there was 36,000 PhDs produced. So, oftentimes this is like people want to pull their hair out and say the sky is falling because there's all these PhDs and nowhere for them to go. That's not quite true, right? As a PhD, you're qualified to do a lot of interesting and exciting things. But kind of the issue here is that the PhDs tend to be trained specifically for those limited number of faculty slots. And their training is kind of geared and oriented towards those slots, and that increases the competition to get those places.
So is that tenure?
These are tenure track positions, yeah.
So it misses the whole world of
sure, underpaid
research,
liberal arts
faculty,
colleges.
and
Yeah.
the whole world of biotech.
Yeah. So like I said, there are lots of other exciting things that PhDs can do.
But they still get good science.
Sometimes.
But have you ever seen that plot of where PhDs get to do science?
I haven't. I mean, I know that PhD employment rate is quite good, right? So, if you have a PhD, you're probably not going to be unemployed.
No, because there's this whole biotech business which is exploded.
Sure. Yeah.
Research faculty or good jobs if people get to be.
Yeah. There are lots of opportunities, but like I said, I think especially if you're a tenure track investigator, it's often also in your own best interest to try to find tenure track positions for your students, because that kind of helps to carry on your research program in various ways that might not exist if they took those other positions. So there is competition for these spots. Besides employment, there's also massive changes in the way that we publish our work. So this is kind of a clever paper that was published about a year ago, where they looked at Web of Science papers. So this is a repository of a bunch of scientific papers. It's no surprise that the number of papers are increasing exponentially. Anybody that tries to keep up with the literature these days will recognize this as being a problem. And they also did this other clever thing, which I'm not totally convinced on, but they tried to map what they called the cognitive extent of the literature. And so what they did was they took the titles of papers, parsed them looking for common phrases, and used those phrases as a way of saying, here's the total number of ideas that exist in the scientific literature. I don't think it's quite a perfect metric, but it's kind of interesting that not only do we have an increasing number of papers, but it's almost like, it's not like we also have an increasing number of ideas at the same rate. We have an increased number of papers while the number of ideas are increasing, but the number of papers per idea seems to be increasing here. And so this is population level, but we can also look at this at an individual level. So this is some data taken collected by Aaron Closet and some of his colleagues. So this is just in computer science, but I have no reason to think that this isn't roughly representative of other fields. So what they're looking at is the number of papers published per person for people in their first 5 years of their tenure track position. And so the different lines are people at different faculty institutions. So the top 20% would be like the upper echelon of faculty institutions, and the bottom 20% would be the bottom echelon of faculty institutions. And in the 70s, even if you're at the top one of the top 20% of universities in the country are basically publishing a paper a year. By 2015, which is when this data roughly stops, that has increased to 6 papers a year. Okay, so I've told you a lot of things that have changed about science in the last few decades, and I think that these changes have some consequences about insofar as the way that we do our science and what we see when we look at our scientific literature. So the system I've just described, we have an increasing number of people writing more publications and doing so with fewer dollars, or at least perhaps that the amount of money is staying flat, but the amount of dollars per person is decreasing. And for a number of positions, that is basically also staying flat. So this is like a hyper-competitive environment where you have fewer resources and people are pressured to produce even more. On top of all this, in the same kind of time period, there's become this widespread adoption of Fisherian or Neyman-Pearson kind of hypothesis testing. De facto requirement that you have to get P's less than 0.05 in order to publish your work. And we are stuck with this communication system that was developed in the mid-20th century. And these things have not changed very much in the last few decades. And I should also point out that there's a bunch of additional issues that I haven't pointed out that are causes and consequences of a lot of these factors that involve kind of the pernicious effects of nepotism, gender and racial stereotypes, and wealth inequality as far as like who gets to do this work and what work gets to be done. And so, people have started to recognize that, like, well, maybe the things that we read in our papers we might see some way in which these kind of ongoing trends are being reflected. So there's been an effort to replicate a bunch of cancer studies in recent years. This work the bulk of it is kind of still ongoing. Some, preliminary results have been released recently. But before this, like kind of large-scale effort, there was a few kind of biotech labs, industry labs that attempted to replicate some of these studies, and the numbers that they released, although very sparse in terms of details, tell us that they're having a hard time replicating the things that they see in the papers. This isn't limited to biosciences. So a year or two ago, they attempted to replicate a bunch of lab experiments in economics and came to the conclusion. Sure.
Question.
Yep.
So it's still, right, when they say they couldn't replicate something,
Yep.
does that usually mean that, that the results are different enough, or that the final conclusions, like instead of saying 60% of something happened, it's 65%. Does that
Right.
count as not replicating, or does that count as replicating?
Yeah, I mean, it's a good question, and different people will come to different conclusions given the same data, right? So unfortunately, this like 6 of 50, the Amgen and the others, they didn't really tell us a lot about what they did. They just basically said, like, we tried 53 times and only 6 replicated. That's all the information we have from those. The cancer studies that are ongoing, I was at a talk about a week ago where the person that's directing that was describing the process that they're going through, and they're increasingly coming to the realization that even with the assistance of the previous investigators, they're not even able to run the experiments because there's not enough detail and not enough information was recorded the first time around. And so, being able to replicate or reproduce it is like far, far down the line as far as they're concerned. As far as like in an ideal world, if they would be able to exactly replicate the exact same procedures and stuff, and then, as the issue you point out, 60 versus 65%, does that mean it replicates? Does that not? Like, I don't know, reasonable people can disagree.
But you have implied that in 1970 I would have replicated better. And is there any data on that? Because you've connected this lack of replication to these structural changes that have occurred over the last 20 years or so. Is there any data? I mean, I feel that my field is as crappy as it's ever been. There are, I don't know that the percent of stuff that was
Right.
poorly done, not well controlled, has really increased. But I only read a fraction of the totals going up, and I probably read a smaller and smaller fraction. So, but is there any data about replicability?
So I will say that some of these like replication attempts that I'm not familiar with other fields outside of psychology and a little bit with economics, but they do attempt to replicate research that was done decades ago, and in those cases they tend to find roughly similar rates, for what that's worth. The story I'm kind of trying to draw out here is that there are pressures that are leading to an increased prevalence of this, as far as whether there are other, there's third variable
But
issues
that
and other.
assumes there is an increased prevalence, and that's what
Well, an
I
increase.
think we all suspect there is, and we all talk about, I mean, it was the early 80s when two people in NIH started pointing out how inconsistent many papers are.
Sure. Yeah.
Including such reputable people as David Baltimore. I forget their names. They were quickly squashed. This is another issue that it's much harder to criticize people now than it was a generation ago. Where
You think it's harder now?
oh, much harder.
Huh. I haven't encountered that
Oh,
attitude
yeah.
before.
In my world, fight meetings used to be much more vigorous. So now it's
Yeah. I mean, I do
maybe
recognize
because
that
there's
as
so much on the line now.
Sure.
We
Yeah.
all appreciate it. If we're too critical, they lose their grant or their
Right. Right.
But,
Right. Yeah.
people
Maybe.
show up at meetings and the paper is out the next day. It
Right.
used to be you go to meetings to approve the work prior to publication. There's a lot of
Sure.
issues.
I mean, ultimately, I don't think that whether it's increased or not.
Well, you
I'm
call
not
that
sure.
consequence. That means something's changed. Structural change, consequence, irreproducibility. It probably is true. I don't know. I've never seen the data.
Right. All right. Well
Moving
then.
forward.
Well then, we'll say it's a hypothesis, and we don't have any data at this point, right? So at any rate, they found about 60% of the economics experiments that they tried to replicate. They replicated it. In psychological science, this is the kind of first effort that they engaged in quite a while ago, and they found about 40% in that case. And then this is unpublished, but this is fairly recently. I encountered the work of Shirley Wang, who's at Harvard Medical School, and she's a pharmacoepidemiologist. Nailed it. So this is a case in which she was using the same data, same analytic procedures, and was attempting to replicate papers in her own discipline, and found that she was able to do it about 63% of the time. Okay.
Yeah, but I'm still struggling with, I mean, there must be some definition of replication.
Yeah, so
There's many definitions is the problem. So you could, so here's some examples. You could
Otherwise, it's
say,
meaningless. Otherwise, I look at that and I don't really know what that means.
sure. So we can scale, I don't want to kind of get too in the weeds about which number we should pick as far as replication, because like I said, reasonable people can disagree. So if you run one study and get, P is less than 0.05, but the next study you get P is 0.06, I don't know. As I said, I'm working in this dichotomous world just kind of for the purposes of communication, but I would say that that's probably good evidence that you're studying the same thing. There are more kind of sophisticated ways of doing this. You can assess confidence interval overlap or something along those lines, or whether it's even in the same direction as the previous work. So there are increasing levels of, I guess, stringency, and as you pick more liberal thresholds, these numbers will increase. So pretty straightforward. But there are.
Sorry, go ahead.
Oh, go ahead.
My impression of the stuff that's been published on this, the drug company that went to replicate a bunch of cancer findings, they said that most of them, I forget the exact number, were replicable. I didn't get the impression they were being very strict, right? Because the drug companies were interested in making money. They weren't trying to make a paper about how science is working. They were trying to find targets that they could take to market.
Now,
I think that by and large, these are not really strict definitions of replicability, but you're right. I mean, there's going to be a lot of judgment calls along the way.
yeah, but I just want to know what they are. I mean,
I think it's going to vary from paper to paper, right? It's going to hard.
Typically in a single paper, they'll report 3 or 4 or 5 different ways of assessing replicability. These are probably about the median marks of the things that they report in the paper. And yeah, I mean, as far as assessing it, that's about.
I don't think it would be objective. I think a human has to make a judgment call at the the end. So you just have to look at that human and decide whether they're doing something reasonable.
Yeah, they also, I think in not this psychology replication paper, but in a subsequent one, they did collect kind of subjective judgments from other researchers and said, here's the results. Do you think this is a replication? And the number is not substantially changed. Okay, so one kind of many different takes on this kind of state of affairs. One, I think, was nicely voiced by Jeff Settleman. So he's at Calico Life Sciences. He pointed out that you can't give me and Julia Child the same recipe and expect an equally good meal. Right? I don't know anything about Jeff Settleman as a cook, but I know Julia Child is presumably very good, and so I'm willing to buy this as far as his cooking skills. But the idea that he's voicing here sounds to me like a hypothesis, right? This is a suggestion that there's some inherent quality in some people or some labs, and they're better able to do science than other people or other labs. That's a hypothesis, and that is possible to test. And so in one of the subsequent kind of so this is just in psychology, but I don't see any reason why we can't at least think that it could probably generalize to other areas. They tested just this question. So what they did was they took 28 studies. They had the protocols peer-reviewed in advance by, in large part, some of the original researchers. Each study was administered to approximately half of 125 different samples. That's 125 different labs engaging in the same set of studies. So for each study, you have roughly 60 some odd labs doing the work. This includes over 15,000 participants from a bunch of different countries and territories. So we have a wide range of different labs trying to replicate the same idea. And if Settleman's idea is right, we should see that there's systematic variation in the labs that are doing these replications, such that some labs are not going to be as able to replicate their work, and other labs are just going to be more naturally inclined to be able to do this. I should also say that these are again, classic and recent, highly cited social psych, generally social psych, also cognitive psych studies. This is not moving. Oh, the number that replicated, for what it's worth, is 54%.
Are these psychometric tests or MRI?
So they tend to be, let's see, you might not be able to read this, and for that I'm sorry, but it's these are trolley dilemma type studies, if you're familiar with that. So moral psychology. I think there's some Kahneman and Tversky stuff in here. And then some more kind of, in my view, preposterous things. Cardinal direction and SES. I'm not sure what exactly that is, but it just sounds unlikely to be true. Priming heat, which is if you prime heat, to what extent do people believe that global warming is true? So there's a range of different things in here. So I know there's a lot going on in this figure. I'm not going to try to oh, sure, go ahead.
Were the labs used in the studies who were doing the replication, were they all social psychology labs, or were they labs of related disciplines?
They were a range of labs. They were all psychology labs, but every psychology lab has different kind of specialty and orientation. So,
Is there any literature sense that some percent not replicating is actually good for the endeavor? Because you did show your graph about new terms.
Sure.
You put value on innovation.
Yeah.
Somehow, and certainly in my experience, oftentimes a paper had an interesting idea, even though the data couldn't be replicated. The act of not replicating it led to other interesting new ideas, which eventually was replicated.
Sure.
So somehow there should be a certain percent of
Right.
not replication.
Yeah.
50-50 doesn't sound bad to me. 10%
Maybe
only
not.
sounds bad to me. Is there any literature on that? I've never
Uh,
seen any.
No,
How much irreproducible results is good?
yeah.
Is the hypothesis.
This is the classic riskiness of science, right? You want to encourage investigators to go out on a limb, even if it might not.
Well, I guess throughout history there are many ideas that were right, which the data at the time may or may not have replicated or even have been acquired.
Yeah.
It's not clear mental. I think Galileo didn't really roll downfalls on milk in the oil drop experiment to say charge. His lab notebook is riddled with
Sure.
bad science to get what he thought was the right answer. There's many, so they thought the idea was so good, and they have to be right, not crazy. You can imagine a lot of crazy people, and so they didn't really bother with getting the experiments right, and
Right.
probably the replication came in a different set of classic experiments. Could be that it was too early to replicate.
There's a
There's
bit of a
lots of
winner's
ways
curse
you
there
can
too,
think of
right? So we remember
replications.
those, but there's also the alchemist and there's a number of other experiments that were bad science and wrong
Yes, but
that we don't talk about.
you'd hate to have eliminated the bad science, good things because of replicating in the short term. In the long term, there has to be replication. But putting too high a bar in the short term could hurt the endeavor overall.
Yeah, I mean, science is a long and winding road, and sometimes, inherently that means false starts. The only thing that I think I would say in regards to most of that is that, if we have tools that we can use to make our science work better, like we should use those tools.
But have you seen any discussion about lack of replication might have a positive component? We always talk about it negatively, especially me. I've always just thought about me. That there may be a positive attribute to
I mean, I think
letting us explore in a way where we don't hold replication as the most important thing.
I know that there's a lot of people certainly talking about the need for science to occasionally be risky, which I think gets at the same idea. But the grant and funding situation mostly precludes that, right? It's hard to ask to do risky work if the people that are holding the purse strings only want to be assured that your work is going to work. And so I think that's kind of a point of tension there. But I'm fully on board that sometimes we should fund things that are crazy and might not work out. Okay, so these are the 26 odd studies. The original, I don't know if you can see this, but the original observed effects is the triangle below it, and then the kind of density plots are the replication attempts. And we can ask the question like, okay, so which gives us more information about whether something is going to be replicated? Knowing who's doing the work or knowing which effect you're studying? If the effect that you're studying tells you more, then that says that indeed people are studying real things in the world, and that if I tell you how to do it, you should be able to get the same set of findings. So they found, here's some quotes from the paper, we found a very strong correlation of samples across effects and a nearly zero correlation of effects across samples. In other words, knowing which effect you're studying tells you a lot more than who's doing the work. 11 of the 28, so they looked to see which of these effects showed heterogeneity, so which were there like wide differences in between labs, and they found that 11 of the 28 effects did show this kind of heterogeneity, but most of those 11 effects that showed big heterogeneity were the ones that had largest overall effect size. In other words, if no effect was observed, that is if it wasn't replicated, there was also very little of heterogeneity among samples. So if somebody finds an effect, then a bunch of other people fail to find the effect, those people are going to be not that variable. But if you're studying something big and everybody else finds a big effect, then there's going to be a wide variation in what people see. So what do we make of all of this that I've just said? I think you can kind of trace it from Adam.
Sorry, that sounded important. Would you say that again? The
Sure.
second part, that is,
So
if someone claims something and people can't replicate it, it doesn't matter who made the claim.
the point I think is that if somebody claims something, the criticism, and then one other person comes along and fails to find that thing. Oftentimes, a criticism is like, well, they're just like not up to snuff. This work tends to suggest that there's not evidence for that, at least in these data. If somebody, the idea that there's these kind of hidden moderators or things that control the outcome of the experiment that are not the things that scientists put in the papers tends to not be borne out in these data.
But the flip side is if there was an effect that people could replicate, then it seemed to correlate with...
If that effect is big, right? So you can be studying small effects and people can reproduce those reliably.
Yeah.
But if the effect is big, then you're going to get wider variation in how labs, in the findings that labs come to. Okay. So this is Adam, of course, and then that's my supervisor before I came to the NIH. Their name's Stacy Sinclair. She's on the faculty at Princeton, and they kind of they didn't actually say these things. This is me putting words in their mouths, just to be clear. But they kind of bound the ends of what I see as like people's typical responses to this situation. On the one hand, Adam is very much of the science is in crisis camp. Sorry, Adam, if that's a mischaracterization of your views. And on the other hand, Stacy is of the mindset that, you know what, science is always in flux, it's always changing, and it always has been changing, and it always will be changing. I am probably somewhere in the middle. I certainly recognize that science is constantly changing, but that doesn't mean that some of these other things aren't actually problems, right? The idea that if I write down what I did in a paper, that you can't, either there's not enough information there for you to do it, or you do exactly what I said and you come to different findings is a little bit troublesome. So what I'm proposing is that we take the idea of the things that open science tells us that maybe are good ideas and view them as technological innovations. If you're working in a highly technical area, oftentimes somebody will come up with some new piece of equipment, some new algorithm, and adoption of that new stuff is pretty rapid usually, if it really works. I think the things that these open science kind of advocates are suggesting that we can use are also technological innovations. And so we should be no less hesitant to use them in our work. And so these things include stuff that's pretty straightforward, like open data, open code, and open materials, larger and more representative sample sizes. And again, I'm going to talk a little bit in detail about larger sample sizes. The use of pre-registration and registered reports. Registered report is where you say what you're going to do. That is the thing that is reviewed by your peers, and then the paper is published regardless of what you find.
Sorry,
By the way,
still lagging. It's all very interesting.
Sure.
Go back because I'm wondering. Back to your other slides. Since I'm with you, I could argue about
this one?
Science is
Stacy.
crisis.
Yeah.
But the interesting data recently that starts altering where you might arbitrarily put yourself is the efficiency of science. There's more and more, that is, per dollar spent, we are accomplishing much less.
Mm-hmm.
If that's true, would make you feel like science is in crisis. Are you up on that? It would have been fun to, pharmaceutical industry, number of approved drugs per
Sure.
dollar spent is going down.
Yeah.
Number of innovations for government spending.
Are you, sorry, I'm not clear. Are you telling me that this is so, or are you asking if this is so?
I'm asking, you've told us two personal views of science, and I'm saying there is an emerging literature.
Sure,
On
yeah.
whether to ask whether science is in crisis or no, it's the same as it's always been, always in flux.
Yeah.
And
I mean,
do you think about that?
yeah,
And
my understanding is that certainly the efficiency of science is going down.
so it would make you feel like we're drifting towards Adam in this. That would be science is in crisis. We spend a lot of money and we don't learn anything new.
I mean,
It's different than in flux where we've always learned something new.
right, yeah. Yeah, that is my reading of what's going on. I'm not as up to date on that kind of work as some of the other things that I'm talking about here. But my take on what's going on is that it's less efficient. Okay, so a number of open science innovations. These disclosure statements are kind of nice. I'm going to come back to them a little later. But this is just a statement in your paper that says, we confirm that for all experiments, we have reported all measures, conditions, data exclusions, and how we determine the sample sizes. And then, of course, you actually do that in the paper. Posting preprints, so your colleagues can see, like, here was the initial draft of the paper. Here's how it changed through the peer review process or through like other revisions. And so you can kind of follow the track of thought over time. These multiverse analyses or specification curves where you effectively try to figure out what is the set of theoretically justified, statistically valid, and non-redundant analyses that you can do to answer your question. You do all of them, and then you present the results of all of them. And so that gives you a sense of like, well, what are different choices in analytic regimes? What do they do to your analyses? I think it's important to publish null findings, to move away from null hypothesis significance testing, and I think there's some good evidence that awarding funding by lottery or modified lottery is a hopeful way of kind of encouraging people to take some risks, and also to reduce the amount of time that people spend reviewing grants and trying to write grants. Now, not all of these are relevant for all types of science. Perhaps you collect sensitive data and you can't release personally identifiable information in the world, or you're studying some extremely specific population, and so representative sample size doesn't work because there's only a very small number of people with that particular condition in the world. And so I'm not saying that everybody should always adopt all of these things all the time, but some of these things can be used to make your science work, I think, a little bit better or be more efficient if that's your preference. And I think generally you can group these things into two kind of categories. So the first are things that I think we can reasonably achieve as individual scientists. So in the 70s, Richard Feynman gave an address at graduation at Caltech, and one of the things that he said that eventually published in this nice essay called Cargo Cult Science, which is great if you've never read it, I encourage it. He said that the first principle is that you must not fool yourself, and you are the easiest person to fool, right? So a lot of these kind of top several things are things that we can put in place to prevent us from fooling ourselves. And they're easily adoptable by individual scientists. These latter few things at the bottom are kind of more institutionalized problems. And so while we can make some moves as individual scientists to do things that address these things tend to require large buy-in from bigger institutions and larger groups of people, and so it's harder on an individual level to do anything to change those. Okay, so I said I would talk about sample sizes as a particular case of this, so I'm going to dig into that a little bit here. I'm cognizant of the time. Is there a point at which I should stop talking?
We'll wait.
In 10 minutes, okay. All right, so if you're working in a regime of measurement uncertainty, it's important to have a large N in order for you to accurately measure whatever effect you're interested in. And so when I say large N, that usually people think of individuals, so participants or rats or whatever. But it could also mean a large number of trials, a large number of countries, a large number of scanners, or a large number of labs or sites. The reason that we want to have large N is because we work, as I said, in this kind of p is less than 0.05 world, and so we want to be able to detect our effect with reasonable fidelity. So if, may have been a little while since intro stats, but probably you all encountered some figure like this at some point. Basically what we have here is on the left we have the null distribution, on the right we have the alternative distribution, and this I've set up a world in which the effect size that we're trying to detect is a Cohen's d of 0.5. Effect size is just a way of standardizing the things that we're studying so that everything is on a common metric. The basic idea is that it's mean of one group minus the mean of another group over the standard deviation. And so it's just a way of making everything kind of on the same ruler. So this is what we're taught to do. You should have a power of at least 80%. That means if you're actually studying something from the alternative distribution, you should detect it 80% of the time. That entails that 20% of the time we will make a mistake such that we will be studying a true effect, but we'll say well, it's not there. And in order to detect a Cohen's d of 0.5, we need a sample size of about 31. But there's been a lot of data and kind of criticism suggesting that we're not typically powered appropriately. So there was this button paper called Power Failure. Power Failure was the kind of snappy version of the title. And they found that in the neurosciences, the power that people were typically working at was about 0.21. And so that's what this looks like. So there's a number of consequences here. The one that I'll just kind of point out is that if you are in fact studying a true effect and you're powered at 0.21, the probability that you're going to falsely conclude that there's nothing there is about 80%. So 4 times out of 5 when you're running your study, you will say well, we didn't find anything, even though you are studying a real thing. And if your Cohen's d, if the effect size that you're studying is 0.5, that means that you're running a study with about 5 people. Probably that's not typically what's going on, that we're like really squashing the number of people. More kind of more favorable view of what happens here is that you do your due diligence, right? You figure out, you run a power analysis, you figure out this is how many people I think that I probably need to detect this effect. And then, so let's say that you figure out that you need about 31 people. The issue is that your power analysis, you're basing that effect size on things that you see out in the literature. And we've already discussed that that's not a representative sample of what happens. Because we have this bright line cutoff, only statistically significant things tend to be in the literature. And so in fact, you think that that effect size that you're targeting is 0.5. We know that the power in the literature is closer to 0.21, and so your effect size is really probably, 0.21, and that means you're about 78% likely to make a false conclusion. So that's the way that power analysis is typically taught, like you're worried about making a false conclusion or a correct conclusion, but there's kind of other ramifications that I think are underappreciated about this whole issue. And so this was articulated nicely by Andrew Gelman a few years ago. This is a slide taken from one of his talks. And in addition to true positives and false positives, he talks about these things called type S errors or type M errors. So a type S error is if you get a statistically significant estimate, what is the probability that it's in the wrong direction? So that would be like a really big problem, right? You think you're studying something positive and you get a statistically significant effect, but it's negative. The other problem is kind of related to what I was talking about with the misspecified sense of how big of an effect you're studying, and that's the type M error, or what he sometimes calls an exaggeration ratio. And that is if the estimate is, so if you run the study and you get a statistically significant estimate, statistically significant effect, how much bigger must it be than reality in order for it to be statistically significant, right? So the reason that that's true is like, here, the true effect is roughly right here, but in order to get a statistically significant effect, you have to be out here. And so that means, like, by definition, your estimate is going to be bigger than what it is in reality. So there was a kind of retake of that power failure paper where, the authors did some good work in saying, neuroscience is a big field. There's like heterogeneity in the field. There's lots of different types of neuroscience. Some of those areas of neuroscience have more severe power issues than others. And so they kind of divided it up, and they did some other kind of interesting stuff, but for our purposes, they divided it up into genetic, treatment studies, psychology studies, imaging studies, neurochemistry, and miscellaneous. And then you can see the number of studies that they used to estimate the median power in that area. These range from miscellaneous, who knows what that is, at 0.57, down to genetic studies, whose power is about 0.11. So that type S error, which is the probability that you're making a mistake in the wrong direction, given these power statistics for a genetic study, 3% of the time, a treatment study, 0.5%, psychology is less than 1%, imaging, one time out of 1,000. So, my reading of that is that this type S error is probably not typically something that you're going to be so concerned about. That doesn't mean that you won't get an estimate in the wrong direction, but it's probably not going to be statistically significant most of the time. The type M error is a much bigger problem, though. Even in the best of these cases, your estimate is still bigger by a factor of 1.3 or so than reality. And so if you're using information in the literature to plan your research and trying to conduct efficient science, this is a really problematic state of affairs. But as I pointed out, N isn't always the be-all, end-all of your work. There are occasions when N isn't as important. So off the top of, this is taken from a paper by Steve Goodman and some of his colleagues a couple of years ago. If you're studying deterministic or near deterministic systems, obviously your power is basically infinite in that case. Or if you're doing something like a proof of principle, right? So think of Dolly the sheep or human limb regeneration. This is the proof of principle idea is, obviously important, but I think Dolly the sheep and human limb regeneration are like exceptional cases of that, right? Those would obviously command a lot of attention by your peers and by the press and yada yada. And if you didn't do it right, people would discover very quickly. But most times, proof of principles, won't be that.
What's the difference between proof of principle in your mind and large effect size?
You need
Proof of principle, or
you only need one observation in order to show that it's a thing. And then provided that, what you said that you did is what you did, you didn't like introduce some confound or whatever. Yeah.
But they're equivalent to large.
It would be equivalent to an infinite effect size. Because even with a large effect size, as long as there's some variance in what you're studying, there is.
To say the live regenerated, you have to make a measurement that had some error. So to say the live regenerated, there's an effect size on that. It's very, very large. It's not infinite. It's large.
Could you apply that to Dolly for me?
Huh?
Could you apply that to Dolly for me, the sheep?
Well, they got to sequence DNA. You've got to decide whether it really arose from the single nucleus you cloned. And there are experimental criteria with errors. So
Could have been contamination in the sample.
Okay, all right.
Your genetic, that's not Mendelian genetics, because the effect size of me carrying a lysosomal storage disease mutation is huge. N equal 1, I can tell. Right?
Right.
So that genetics is a funny term there. There's many things we do that are not proof of principle, or that merges large
Sure.
equals.
Yeah, I mean, I am all about the gray areas of everything. I kind of,
Well, this is another example too. I mean, there was in the early days of fMRI, there was, demonstrations of like retinopathy. You could do retinopathy, and there were probably anywhere from 5 to 10 subjects. And just they're all kind of similar, and that was enough. Everyone was convinced that you could do retinopathy. So it's sort of like it's somewhere between a proof of principle and there were no statistics on like what consisted of retinopathy. How we are we certain that it's retinopathy, or we weren't asking any sort of second level questions. Just
Right.
Can we do retinopathy?
Yeah. Yeah, I mean, I'm willing to grant that there are definitely kind of gray areas here. I will point out one kind of interesting discussion of this. So I found this paper, Does Chemical Engineering Have a Reproducibility Problem? So this is a field where they're trying to make new materials, right? And so effectively an N of 1 is sufficient. But there are lots of times when people make new materials and then that's it. Nobody follows up on it. It's not so important. So there's this kind of gray area. I was gonna get into the details of this, but I'm gonna skip it for now and just point out the fact that if I were to kind of answer this question, does chemical research have a reproducibility problem? First of all, I'm not really qualified. I'm not a chemical engineer. But I would say if they were worried, there are things that they could do to make their, technical innovations to make their science better. In particular, I imagine a disclosure statement on steroids. So something, anything that imaginable under the sun that you think could have influenced your results, you make sure it goes in the paper. And then, if you really want to go above and beyond, mandatory independent replication of whatever you've done by another lab as a precondition for having a publication. So I think, that is a technological innovation that is practical and doable. Of course, it is time consuming, but, if you think what you're studying is important, then that might be worth doing.
So I think that gets to an interesting point, if I can interrupt just for a second. So yeah, you can imagine there would have been a mistake with Dolly. Maybe there was contamination in the sample, or that it's harder to imagine in the liver generation or in retinopathy that maybe there was some sort of problem with equipment that wasn't real. So we think about these things as that we just believe them after the fact, but it's definitely the case that somebody else tried it later, right? So we also have cold fusion, right? And a number of other things where somebody tried it. There's the stem cell thing that happened in Japan where somebody tried it and said, no, this is not actually replicable. So if we sort of built in replications as what you're saying for the publication, not for the preprint, absolutely. I mean, I think that we should encourage people to be out there and exploring and doing things like the guys who did the gravity waves, right? They actually called up 3 different labs saying, we see this crazy thing. Can you see it from your facility as well? If we change the incentives in science to encourage people to do that extra set of replication, we'll get a little bit more efficiency, I think is the idea.
Well, the incentives are quite high. If you do consistently good work, we all know it, that you get, somehow you get credit for that. If you do consistently poor work, we all know that too.
Well, the other thing that Travis showed to argue that's not the case, that it's not about the careful labs. The careful labs can put up crap just as well as anybody else can. It's just you need to have some sort of,
I'm not sure he did tell us that. That's why I was trying to figure out what that study did. That's right. So,
I mean, I think we should be careful to, this idea that some people are like naturally better or perhaps have better training to produce the same kind of work is not something that, I think that one particular study demonstrates that probably there's at least some evidence that that's unlikely to be true. And furthermore, for a given person, even if you are willing to accept the premise that some people are naturally better than others at producing this kind of work, that doesn't mean that they are consistently naturally better. That just means that sometimes they tend to produce really great work. Sometimes they might produce crap. And I think, it's almost nonsensical to not take advantage of these systems that prevent you from producing stuff that is not likely to be true. I will say that the only reason why we might not do it is that these things are time consuming. And if you want to have a career in science, you need to publish a ton of papers and you need to do a lot of work. These things deliberately slow you down. I think that's maybe a good thing.
but you have told us there's such a thing called proof of principle.
Sure.
And there are some environments that really value that. I would say that's a large effect result. And somehow the effect size, you can't lump all this together. You already have distinguished a class of work that's different. You called it proof of principle. I would call it large effect size. A radiologist diagnoses a brain tumor. N=1, you've got a brain tumor. That's not proof of principle. That's diagnostic. It's done every day, all around the world, all the time. Effect size is about 6 on that call. If you get GAD in an MRI scanner to make that call. So, to do this without some recognition that some environments value a large effect size or discovery of new things,
Sure.
principle, and those are the people we tend to value more, I would say. Maybe that's not true, that's another hypothesis.
Well, that's like a disciplinary difference, no?
Huh?
That's like a disciplinary difference, not like a within discipline which things are valued. That's this particular discipline is.
No, I definitely know fMRI environments that have a much different attitude towards the data and what they're hunting for than other fMRI environments, which barely know what they're doing. They use CAN sequences and CAN processing and are applying it compared to, there's a huge range of fMRI environments, and some people consistently do things well. Others, next week they're doing something different, and next week they're doing something different, and next week they're doing something different. They're consistently not really contributing. So it's crazy to deny that there's a difference in quality from lab to lab to lab to lab. That's
I would agree with that. But so I think you could call it Dolly the sheep, you could call it cold diffusion, you could call the radiology tumor, you could call all of those things large effect sizes. But some of them were wrong. Stem cells are the pluripotent stem cell thing, and they were not replicable, right? And you would say
people make mistakes.
Right. But I think that's what
those can't be replicated, but you are very careful to sort of eliminate, bad mistakes.
But in the cases of pluripotent stem cells and cold fusion, within a month, the field had gone out and tried to replicate it and demonstrated that it wasn't. I would argue that there are thousands of things that are not as important as cold fusion or pluripotent stem cells that are in the literature that were done with some kind of error that meant that they're not real, but we think they are because we read the paper.
Well, you may think they are, but that's your decision. There's a lot of stuff I read, peer review, and I walk away with a quick little physics calculation and say it couldn't be true. How did they get through peer review? We're all free to evaluate the literature.
But at the end,
as you know, I look at effect size.
Right.
If it drops below 0.5, I ignore it. Literally, I ignore it. Not that it won't reproduce, and I'll just wait. But other people work in that area, right?
But there are, I've seen it happen a number of times where there's been something that was kind of sexy in the literature, and dozens of labs
Yes,
were
wasted
trying to
their time.
reproduce it, right? And it wasn't until a decade or two later that we said, oh yeah, that was wrong, right? After we'd
But
spent.
there are probably dozens of other labs who read it and decided not to do it.
It's
I mean, what can you do?
But you can imagine an environment where the funders and the publishers said, you know what, we're not going to put that out. Why don't you go find another lab to do the exact same thing before we put that in Nature or Science? Right? We could do that. The NIH
Well,
could do
the
that.
GWAS community, when they realized how much trouble they were having replicating, put a very high bar for replication. You know,
After a decade of
4,000, go get another 4,000 and make sure, you know, the associations hold up. So there is a community that has a very high, they funded it too.
They have figured it out. I don't know that the rest of us have.
So
Sorry,
but-
stand past up such a late
But really quickly,
I mean, that's
sorry.
basically it.
I mean, is this a crisis? I mean, it still seems to me that there shouldn't necessarily be any top-down control over this. I mean, we're doing what we can, but I mean, it seems like the field is very much self-filtering with a lot of accuracy over time.
I mean, I would be hesitant to suggest that most of these things should be top-down implemented. What I think is more realistic is that people will read papers that have open data, open code, blah, 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 and they will give more like subjective value to those papers. They will perceive those papers to be stronger and better. There are some of these things that it's hard for us to do independently. So for instance, awarding funding by a modified lottery, like that is not something that I as a scientist can decide like, oh, I'm just gonna give my like, you know, $20 in my pocket to whoever by lottery. And so there are some things that I think are institutionally required changes or, you know, could be beneficial. But, you know, many of these things you can decide to do independently or not. But, you know, at the end of the day, it's like you have to consider what adopting or not adopting these things is going to do to the quality and productivity of your science. And I think one of the problems is that I think those two things are in conflict.
Well, it's tricky because it's already we have mechanisms set up like, you know, with medical research. You know, if it's truly potentially useful, like it's, you know, there's FDA, you know, trials and things like that. So it sort of gets tested, even if, you know, those are flawed as well. But I mean, the fact is nothing is perfect, but ultimately it gets winnowed out. You know, ultimately things that actually work get in there.
Yeah.
It seems right.
Well, unless the efficiency is going way down.
Right, deficiency.
I mean,
right.
if we are wasting a lot of money and the place we're wasting it is on bad science,
Yeah.
that's really
Like
Yeah.
puts us in a
Right.
crisis.
So there are things that we could do to make it slower and more careful. And I think there's an overwhelming evidence that we should do that. I agree, science will eventually sort it out, right? But we could sort it out in 2 years instead of 20.
Okay, so the way I think of this, there's kind of two ways of thinking about scientific literature. There's some people who like to think that scientific literature, nothing should be admitted unless it's absolutely true, and the patent's body is absolutely true to life. And another one that says it's a way of communicating to other labs and scientists. And so when you get some results that are interesting, you have what you believe is a sufficient amount of data, you can put it out there, and then that's kind of more like an invitation to discuss. Okay?
Preprint.
And so,
That's what the
so you
preprint
say, for example, that you should, before you publish, you should maybe go get somebody else to reproduce the work. Well, perhaps a publication is the invitation of having somebody else do the work. Do that with verification. It is interesting, yeah.
The thing about that, well, I agree that, like, yeah, putting something out there, if you think it seems interesting, other people should reproduce it. With the way science stands and funding stands, people are going to reproduce it. That is not what gets you grant money. That is not the studies that are going to get published unless you say this study was wrong. And people are not going to take the time to do that because they are not going to be able to keep their lab going. They're not going to be able to get the money for science that way.
Unless there's RFAs for reproducing.
Yeah, but there's two truths to this. I mean, so it used to be, certainly those of you who read papers from the 70s and 80s, like it used to be that as you wrote your paper, you said, hey, these people found these things, and I want to study this thing that's kind of the extension or along the way, midpoint of these things. And they would publish sort of the, like, so dyslexia and checkerboards. Like, this was a huge thing. It's like, present checkerboards to people who have dyslexia. Are there differences in the ERPs? And at first it was yes, and then it was no, and then it was maybe, and then it was definitely no. And I see this trend republished every 10 years because everyone forgets to go back and reread the old things. I think what makes Andy's argument particularly interesting is that instead of shifting to preprints, what we could do is ask for a next level of journals, which is the journals that combine multiple things together. Not necessarily meta-analysis, which is another way of doing this, but perhaps a narrow image with replication journal that suddenly says, in order to be published here, you have to get 2 independent groups, or you have to do this on 3 samples. So perhaps it's not necessarily, I'm throwing this out there, you're scowling at me.
No, I'm not.
That's right. But perhaps there is this other mechanism that we could push for, which is higher level, even more elite journals that then give people, as they always have, better chances of getting grant funding.
I've seen this suggested such that journals should be split into 2, and there's one section of the journal that is like, here's the crazy stuff that might be true. We don't really know, but it's interesting. And then there's another section of the journal devoted to like, no, this is absolutely true. And if you kind of you allocate kind of credibility and belief and respect to papers that appear in different sections accordingly. And that, I think, is exactly what Pete's kind of suggesting here.
I suggested to previous NIS director, Story Landis, who launched this rigor thing. Now we all have to make sure you don't have all of that on the list. What's inclusion, exclusion criteria? Do you really know what reagents you're using, et cetera, et cetera, et cetera? People sit down an MRI scanner, they think the pulse sequence worked. Some places do a great job on making sure those pulse sequences are running regularly. Others don't know what the hell their pulse sequences are doing, as an example, in the MR. I think, why don't you give a prize, a really nice big prize for someone who's had a whole career of boring but reproducible work in a lab, right? Because they look for the stars rather than the foot soldiers. In the end, it's right, how to give incentive to doing high quality, but that assumes individuals do high quality work, which you told me no, it's shocking.
It's slightly different.
Yeah, and certainly NIH grant fundings and the extramural programs sort of reflected this like high risk versus true science approach for a while, right? So you had R03s, which were sort of like small proof of concepts grants. You had R21s, which were like more money, 425,000 over 2 years, that were like high risk, high reward. And then you had the R01s, which were up to 5 years and millions of dollars, that were supposed to be based on steady science. But of course, the problem is that the caps on those grants have gone up over time. And so people start to say, I need to pay for MRI time, which is $500 a scan. I need to pay for a methodologist and these people. And so they're trying, I think, to sell more things as tried and true science. And everyone kind of acknowledges that R01s have become the new high risk, high reward. And R21s are just as much work as an R01. And so fewer people apply for them. Which is part of the reason why the funding cap, or the funding percentile for R21s is, at least recently, was higher.
Do you think Story's rigor thing worked? I feel like you feel like it's mostly wasted time. Do you think that?
There's not enough emphasis on effect size in mine. That is, in the end, while it's true, small effects can be replicated. It's just very hard. You have to, so I think all this is great, except why not teach us all that, in general, big effects, if you do assuming the experiments are being done without artifact, they're more interesting, they're more exciting, they're more useful, they change the world. Small effects are hard, and that's, everybody writes that in a paragraph early in these discussions, and then they go on to trying to fix small effect science. And I don't know what's going to fix small, because the distribution. I mean, they have to have large N's, but epidemiology is a field filled with large N's and often has trouble replicating because they're small effects, right?
What do you imagine, if you could imagine the distribution of effect sizes in the population of all things, what do you think that distribution would look like?
Say it again?
Like, what do you think the distribution of effect sizes in the population looks like? Do you think there are a lot of large effects that we just need to find, or do you think there are vanishingly small of them? And most effects are small.
We barely understand anything, so if understanding something new is proof of principle, large effect, we have a long way to go. The best symptom of that is we have only annotated a small fraction of the E. coli genome. I think 30% or something, right? So in the case where we know there's stuff being made and that stuff probably has a function, 2/3 of the E. coli genome, we still don't know its function. Yeast is even worse. Us, it's unbelievably so. As an indicator, we have a huge amount to learn still.
And
In your sense of proof of principle, what the hell does that thing do? Can we all agree on that? Yeah. So yeah, that's hunting for large effect sizes and optimism that we still have a lot to learn. If you feel like we know everything and the thing to do is to put it all in context, then that's a small effect world. It's very pessimistic if you ask me. But
do you think that most scientists are looking for these large effects, or do you think most scientists
a
are?
lot on the field, the environment.
Why is that?
Huh?
Why? I said, why do those things matter about what kinds of things people are looking for?
Oh, because there are things that are interesting
To
that have
whom?
attracted people, even though the thing they're studying tends to have small effects.
So,
And they should be able to study those things. I mean, I'm with Peter. You don't want to top down and tell people don't follow what you're most interested in.
so but okay, so yes, there's definitely a lot of things that are going to need to be discovered, but we don't have the methods or don't have the ideas yet. And it's a long path to get there. But what I worry about is if you tell scientists to only go after large effects, or that you somehow incentivize large effect science, I feel like there's just a lot of boring work to be done to figure out how different systems interact. And they're going to interact in relatively small, mundane ways, right? That are not going to be large effects, right? I guess I feel like we need to incentivize scientists to be careful and to spend a lot of time in the boring details, and that eventually we will get to the big effects. So
what's the evidence that when you do these large end studies that they're actually done really carefully? Because you have been participating in discussions where you're talking about some very large scale studies where there are all sorts of methodological decisions made with very little consideration, right? So yes, you're going to collect 10,000 subjects, and whether or not you have chosen your parameters with any degree of openness, clarity, or proof, it's not there.
It always tend towards not replicable.
Well, it may be replicable, but it may be crappy.
Right.
Or you may have large data sets where people say people are in a resting state. Oh, maybe 20% or 30% of the time they're actually sleeping. Oh, we didn't control for the behavior. But we have a lot of those subjects in uncontrolled states. But we have very large N.
large N does not fix everything.
Large effect when you're sleeping. Sure, no way. And there are people who are going and mining that data to determine exactly when they were sleeping, but they don't actually necessarily have all the data to be able to do that. I mean,
I think Google is the one who argues that if you have enough data, at some point the messy stuff kind of washes out. There are certainly cases where that's not the case.
anyways.
I don't have anything else left. I just wanted to thank everybody here who's welcomed me and talked to me about these ideas and some of the others. That's the machine learning team and the data science and sharing team, members past and present.
Thanks, Travis.
I think another interesting distinction is rigor has made it. It's called it exploratory. Yeah, there's an environment where you're meant to be exploratory and an environment where you really are